Hey everyone, so I'm making this video as an introduction to the next part of the class, and what we're talking about is going to be um, sort of the idea in biology. I couldn't emphasize it enough saying, this is the deal. And in biotech, this is what we're trying to manipulate. So having a solid understanding of what's going on here really helps you with what's going on in terms of all the manipulations that we do in biotechnology. And it really revolves around these three things. And you guys might remember when we talked about how nature inspires us, and I gave you several examples and some notes about how these three things are connected, right? Where a phenotype is just the physical expression of a gene. And the middleman between those things is something called a protein. So all of the things that we look at in biotech are really based around proteins. Like proteins do have specific shapes and they do really specific jobs, and those are ultimately coded for by a gene. So there's this relationship between these three things where, if we have a change in a gene, it's likely that we'll have a change in a phenotype because we had a change in a protein, right? That's how this flow of information all works together. So what I want to do is give you a nice kickoff to the next set of things that you're learning about, but let's also look at what you just learned about. And what you just learned about was the structure of DNA. And there's certain things that are going to be really helpful for you in this course to understand, and also understand this next idea if you understand these parts about DNA structure. Okay, so this is what you should understand. Number one, the DNA is composed of nucleotides, and that nucleotides are essentially made of those three separate components that we talked about. They're made of a phosphate group, a sugar group, and they're made of a nitrogenous base, A, C, T, or G, right? At least in DNA. You should also know that DNA is anti-parallel, meaning that it has directionality, right? And we mark those directions by something called 5' and 3'. And when we say anti-parallel, that means our DNA strands, they are parallel to each other, but they're anti-parallel because they're flopped. So in one strand, we're going to run 5 to 3, and the complementary strand is going to be 3 to 5, so that those bases can hydrogen bond with each other. You should also understand those bonds involved in the structure, right? That there are hydrogen bonds that interact and link these nitrogenous bases together to form the double-stranded molecule. And then the things that join the sugar and the phosphate all along this backbone right here are called phosphodiester bonds. It's what links the DNA together and ultimately gives it its structure. And DNA has two complementary strands. A always pairs with T, G always pairs with C. Between A and T, you have 2 hydrogen bonds, G and C, you have 3 hydrogen bonds. And again, we'll take advantage of all these things in biotech. And those are really the main things that I want you to know for one. We can do all sorts of really cool stuff if we just know sort of those fundamentals. Now, let's get into the new stuff, right? This is just a reminder, but it's a pertinent reminder, and it's something that you'll look at specifically here, of examples of the relationships between what I started this video with, phenotype, protein, and genes. And it will lead to what our big question is. So let's just focus on a phenotype and a natural phenomenon. Something that we can notice is lightning bugs. And unfortunately, we don't have those in Colorado. But if you've ever seen them, you know that they exhibit a glow or a fluorescence, a bioluminescence. They light up, and that's going to be their phenotype, right? That phenotype of being able to light up is ultimately controlled by a protein that does a really specific job, and that protein is called luciferase. What it does is it binds to a molecule called luciferin, interacts with some oxygen, and it gives off light. In part of that chemical reaction, it gives off light. So that protein, this purple glob, is what's ultimately responsible for giving the phenotype. And that protein is coded for by a gene. And genes are a sequence of A's and C's and T's and G's. Okay. Another example is with lactose intolerance. The phenotype is something like cramping and bloating, discomfort that a lot of people have when they have lactose intolerance. That's the phenotype. That's ultimately controlled for by a protein, and this protein's name is lactase. What it does is it breaks down the sugar in milk called lactose into its component units. It breaks it down into glucose and galactose. If that protein isn't working, then you're going to have the phenotype of lactose intolerance, right? That protein, you guessed it, is coded for by a gene, a series of A's, C's, T's, and G's. So I think at this point it's helpful to define what a gene is. A gene is a sequence of DNA, A's, C's, T's, and G's, DNA nucleotides that code for a protein. And that ultimately is going to lead us to our question, what is the next thing that we're going to look at? And that question is, how exactly does nature turn all of that DNA into the flesh and blood of you? Or another way of asking this question is, how are genes, how do they code for proteins? And what's the process of going from code or a gene to a protein? What does that look like? Right? And that process ultimately is called protein synthesis. Like, duh, we're making proteins. That's what protein synthesis means, right? The synthesis of proteins. We're making them. Ultimately, that's going to come from the DNA code. But all the DNA code is good for, remember, it has to stay stable. It doesn't do anything interesting. All the interesting stuff is done by proteins. But DNA is the code for how to make those proteins. And there are two separate processes that make these proteins. One is called transcription. And in transcription, what you'll see is that DNA is copied into another molecule called RNA. And then in the second stage, called translation, that RNA is read and proteins are synthesized. So ultimately, that's how DNA or genes are connected to proteins, right? So what we're going to do is look into some exploration of how we can manipulate these things, but also how it works. We need to have a basic understanding of how this process works before we can manipulate it. And that's what you're going to begin today. So good luck. We'll go over this the next couple of weeks. Man, this opens up the door. It blows the doors open to all sorts of fascinating things that we can do in biotech.